First is to centrifuge the EDTA samples. Then, transfer the plasma to the other tube. Then, add as much volume of NSS until the mixture fills 2/3 of the tube. Then, mix or gently invert the tube. Repeat the procedure for the other tube. Then, centrifuge the sample for 2 minutes at 3,400 RPM or at speed of 10. Using a pipette, discard the supernatant on the sink. Then, repeat steps 2 to 5 up to 3 times. To prepare a 5% red cell suspension, transfer 3,800 microliter of normal saline solution onto the test tubes. Then, transfer 200 microliter of washed red cells from the previous procedures. Then, mix it thoroughly. A proper red cell suspension has a tomato red color. Place 1 drop of anti-A, anti-B, and anti-D on a clean slide. Then, add to each drop of reagent a 1 drop of red cell suspension. Then mix it using a applicator stick. Gently tilt the slide from side to side continuously for up to 2 minutes. Lastly, read, interpret, and record the result. For red cells, place 1 drop of anti-A, anti-B, and anti-D in a separate clean test tube. Then, add 1 drop of red cell suspension. For plasma, add 2 drops each of serum or plasma to the test tubes. Then, add 1 drop of A reagent red cells and B reagent red cells. Then, gently mix the contents of the tube and centrifuge for 15 seconds at 3,400 RPM. After centrifugating, read, interpret, and record the results. Dispense 1 drop of red cell suspension. Then wash each tube 3 times with normal saline solution. Then decant. Immediately add 1 drop of anti-human globulin to the cell suspension. Then centrifuge for 30 seconds at 3,400 rpm. Lastly, examine the cells for agglutination. Add 2 drops of patient serum to the test tube. Then, add 1 drop of 5% group O cells red cell suspension. Then lastly, add 2 drops of LISS. Then, incubate for 10 to 15 minutes at 37 degrees Celsius. Centrifuge the tube for 30 seconds at 3,400 rpm. After centrifugation, resuspend the cell pellet by gently tipping and rolling the tube. Wash each tube 3 times with normal saline solution. And completely decant the final wash. Add 2 drops of AHG to the dry red cell bottom and mix well. Repeat steps 4 to 5. Then lastly, examine the cells for agglutination. Mix 2 drops of patient's plasma or serum with 1 drop of 2 to 5% donor's red cell suspension. Then add 2 drops of LISS and mix it. Then incubate at 37 degrees Celsius for 10 to 15 minutes. Centrifuge for 15 seconds at 3,400 rpm. Observe for hemolysis and agglutination by gently resuspending the red cell bottom. Wash the red cells 3 times with saline and completely decant the final wash. After washing 3 times, add 2 drops of AHG to the dry red cell bottom and mix well. Centrifuge for 15 seconds at 3,400 rpm and observe for agglutination. Dilute 50 microliters of donor's red cell with 1 ml of DG gel salt. Dispense 10 microliters of donor's red cell suspension to the corresponding well. Then dispense 25 microliters of the patient's serum or plasma into the same well. Incubate for 15 minutes. Then centrifuge for 9 minutes. Then read and interpret results. Prepare the materials needed. Then prick the patient's finger. Then allow a droplet of blood to fall into the copper sulfate solution. Observe if the blood sinks or floats